All right. Well, thank you all for attending this webinar. I'm Mark Sadie, co-founder and chief technology officer of Pathomics.io, a contract research organization specializing in end-to-end analysis of digital pathology datasets. So as a high-level overview, image analysis is the process of applying computer algorithms to process and interpret digital images of tissue samples in a quantitative manner. It's by no means a one-size-fits-all solution. At the start of your imaging study, it is important to consider the methods that may need to be developed to extract the results that you're looking for. And in this presentation, we will review a multiplexed image analysis workflow that we've developed and highlight some practical applications in several peer-reviewed studies. So multiplexed immunohistochemistry is a technique that allows for the simultaneous detection and visualization of multiple targets, such as proteins, within tissue sections using distinct fluorescent labels. By enabling the analysis of multiple markers in the same tissue context, it provides a more comprehensive insight into the spatial relationships and interactions between different cell types or molecules. This method is somewhat particularly useful in research and diagnostic applications, such as studying tumor microenvironments or identifying complex immune responses, essentially enhancing our understanding of disease mechanisms and aiding in the development of targeted therapies. So it's easy to get overwhelmed with the volume of data generated from multiplexed IHC studies, and attempting to visualize all markers simultaneously won't really yield any meaningful information as shown here. But by meticulously designing a multiplex workflow to profile specific combinations of markers and cell types, you can address and evaluate distinct spatial relationships between cells in their native spatial context. And some combinations can include proliferating tumor cells, activated T cells, macrophages associated with immune suppression, and regulatory T cells, to name a few. In this webinar, we will walk you through an example workflow from a recent project done in collaboration with the team at Fortis, and then show the practical application of such workflows from our published studies. First, we will review a multiplex analysis workflow developed for a recent project done in collaboration with Fortis, using open source tools such as QuPath for the image analysis, and Python for the downstream statistical analysis and data visualization. So, common to nearly all digital pathology workflows is cell segmentation. Cell segmentation is the process of digitally isolating and identifying individual cells within tissue images for detailed analysis. Historically, watershed segmentation has been used where the image is treated as a sort of topographic surface, simulating how water would fill different areas to find and outline each cell. Non-AI approaches such as watershed face considerable challenges in achieving accurate segmentations across all cell types. Variations in things like nuclear size, contrast to background, cell morphologies, all of these can impede the accuracy and reliability of this kind of segmentation. Advances in AI-based methods like Stardust outperform traditional techniques by using large training datasets to enhance segmentation precision. Stardust segmentation uses deep learning to accurately outline cells in crowded or complex images, handling overlapping and irregular shapes better than traditional methods such as watershed. There are multiple approaches to take towards phenotyping cells. Supervised classification is done in tandem with the guidance of a subject matter expert, such as a pathologist. Unsupervised approaches autonomously cluster cells to reveal distinct subpopulations and uncover new cellular insights without prior labeling. Using these approaches in tandem can actually maximize the insight derived from multiplex datasets, effectively balancing expert accuracy with algorithmic discovery. But in this presentation, we will focus primarily on supervised approaches and how this can be used to query specific cell types of interest. And the first step is thresholding our positive population, typically done in conjunction with a pathologist. Here, we showed this for several markers in our panel, such as CD8A, pCNA, and cytokeratin. So now that we've classified each cell for every marker, we need to combine these into a multiplex classifier. And by applying each of our pathologist supervised classifiers sequentially, we can then get a list for each cell containing the markers that it is positive for. Cells can then be filtered from this to look at specific subsets of interest defined by more than one marker. We can identify cellular subsets such as regulatory T cells, activated T cells, proliferating malignant epithelial cells, and any other cell types definable by the markers included in the panel. Once defined, we can score tissues not only for single positive populations, but also the subsets defined by more than one markers of interest. In a fundamental sense, spatial biology involves techniques to simultaneously visualize and analyze multiple markers within their native spatial context. But up until now in this presentation, the spatial dimension hasn't really been utilized to its full potential. Area segmentation is one approach to analyze cells in relation to larger elements of the tissue microenvironment. Here, our tissue is annotated for representative tumor and stroma regions using cytokeratin. AI models are then custom trained to find these structures across all images in an experiment. We can then take our area segmentations and combine them with our classified and segmented cells. And this allows us to compute the distance of these cells to the nearest tumor stroma boundary. This can then be leveraged to identify subpopulations of cells immediately within regions such as the tumor mass as a means of characterizing phenomena such as immune infiltration. Some initial spatial features we can generate include cell distance analysis, which is the distance of each cell to the nearest pCNA positive cell, or Delaunay triangulation, which allows us to identify spatial neighborhoods. And while spatial analysis allows us to generate spatial features, what are the practical applications? So in collaboration with nephrologists at the Mayo Clinic, our aim was to use multiplexed imaging to characterize the heterogeneity and identify biomarkers of kidney injury caused by immune checkpoint inhibitors. Immune checkpoint inhibitors are commonly used to treat a wide variety of cancers, but one major side effect is acute kidney injury hypothesized to be caused by the elevated systemic circulation of these immune cells. Prior studies have yielded a panel of potential biomarkers indicative of acute kidney injury caused by immune checkpoint inhibitors, and having the ability to characterize the immune landscape using these markers can actually identify the biomarkers relevant to differentiating acute kidney injury caused by immune checkpoint inhibitors relative to other causes, and fundamentally yielding a more promising prognosis for the patient. So as previously shown, we can segment our cells and begin visualizing marker expression patterns across different disease categories of interest. Independent of cell classifications, we can visualize marker intensity distributions grouped by the cause of kidney injury, termed code. Shown here is simply the mean CD3 intensity of all cells grouped by AKI code. And visually, it may appear as though there is no discernible difference, and that's because the intensities of the true negative cells typically outweigh those of the true positive cells, especially when viewing cell types commonly present in low abundances. However, by using our pathologist-supervised thresholding approach, we can isolate the positive population and score samples accordingly. So by applying the pathologist-supervised thresholds, we can then identify the positive population of cells and find the percentage of cells positive for individual markers, as shown here for CD3. That being said, many immune cell types are characterized by more than one markers. As each cell in the dataset contains a list of markers that it is positive for, we can query specific immune cell subsets. Here, we measured the percentage of CD4 memory T cells as those that are defined as being CD3, CD4, and CD45RO triple positive. Tying this back to the original study, we scored our samples for a variety of immune cell subtypes to identify those that exhibit differential scoring for one AKI classification relative to others. And while many subtypes do exhibit variation, CD4 memory T cells, T helper cells, and dendritic cells were ones observed to be statistically significant. But as the old saying goes, seeing is believing. So it's important to validate that what our results show is consistent with the images themselves. Using additive color theory, we highlighted the relevant immune cell subtypes in each row using representative samples of AKI classifications as columns. In the top row, CD4 memory T cells were defined as those that are CD45RO, CD4, and CD3 triple positive. And given that each single positive cell type was labeled with a primary color, the triple positive population is shown in white. Similarly, T helper cells, shown in the middle row, were defined as CD4, CD3 double positive, and highlighted in cyan. And dendritic cells, defined as HLA-DR and CD11C double positive, were shown in yellow. So this pathologist-in-the-loop methodology for cell classification can be extended to additional studies that necessitate a comprehensive analysis of the spatial distribution of these immune cells. And on that note, let's discuss one application of spatial biology in characterizing the immune landscape of kidney transplant rejection. So more often than not, there is an underlying notion that once an individual receives a transplanted kidney, that they're on the road for recovery. In reality, many complications can arise. In fact, up to 15% of transplanted kidneys will exhibit signs of rejection during the first year of transplant. And identifying the cause of the rejection can actually enable clinicians to treat the rejection before it progresses and requires removal of diseased tissue. So in collaboration with the Mayo Clinic, we received 247 multiplexed images from 32 archived patient samples, bearing a confirmed diagnosis from one of 6 BAMF classifications of renal transplant rejection. Our objective was to develop a method to characterize heterogeneity within and across patients and explore how the spatial dimension can yield a more accurate stratification. So in this study, we will demonstrate the pivotal roles played by each step of the analysis as previously outlined in this presentation. Just as previously shown, we can score samples grouped by a categorical variable such as disease classification. We can quantify the percentage of cells positive for an individual marker and visualize these results as a heat map. And while this does allow for the simultaneous visualization of several markers in tandem, we do lose a bit of information regarding the distribution of scores from samples across our population. With box plots, however, we can actually view individual samples as points and metrics of their distribution, such as median, interquartile range, and outliers. Here, we show the scores for CD4 memory T cells, again as being defined as CD3, CD4, and CD45RO triple positive. But at this stage, we've yet to really take into account any aspect of the spatial dimension, such as spatial localization of cell types to various renal structures. Here, we display the relevant markers for renal structure segmentation, such that a pathologist can annotate their structures of interest. In this study, we were interested in the localization of various immune cells to glomeruli, tubules, arteries, and interstitial areas. And after obtaining over 3,100 annotations from our pathologist, we then trained a pixel classifier and applied it to all images in the dataset. Several different classifiers were used, and we varied the type of model, convolutions, feature scales, and markers used. But ultimately, we opted for the model with the highest overall accuracy and a blinded validation set. And much like how lymphocytes infiltrating into a tumor mass is more indicative of cancer patient prognoses than cell counts alone, the renal localization of immune cells can be indicative of rejection cause. We can apply the same percent positive scoring approach to explore differential localization of various immune markers and immune cell subtypes to glomeruli, arteries, interstitium, and tubules, with a representative image shown above and the structure highlighted in yellow. The main strength of this approach is that it guides cell type discrimination through the eyes of an expert observer. But we'd also like to investigate spatial features associated with the heterogeneity we've observed across our patients. So as part of this study, we have created a framework for predicting categorical variables from supplementary sample categories. And this isn't just limited to kidney transplant rejection categories. This can also be applied to any feature of a dataset in which comparisons across one or more groups is needed, such as differentiating a control from experimental treatment category. Once the model has been trained, we can identify the spatial features that play the most important role in classifying these patient samples. Much like we had done with the percent positive scoring heat maps, we can visualize the most available features for the classifier, grouped by rejection category. But as previously mentioned, heat maps lack the ability to portray population distributions, and when visualized as a violin plot, we can truly appreciate the heterogeneity in feature expression across disease types. This indicates that it's not always just one feature actively differentiating a disease, but rather multiple in tandem. But again, as the old saying goes, seeing is believing. So here, we randomly sampled 40 regions across our samples with the raw EK-adherent channel shown along the DNA counterstain. Next, we show an overlay of a non-spatial feature, in this case, mean EK-adherent intensity, across a whole cell. And you can see that there is some variation across our samples. But now, if we visualize the fifth most important feature in our model, a greater degree of inter- and intra-sample heterogeneity can be seen. But how does this fare in practice when trying to resolve two similar disease categories? Well, historically, chronic and acute antibody-mediated rejection have been quite a challenge to differentiate. And while variable expression of EK-adherent has been observed in the past, the difference here is quite minor. But when viewing our fifth most important feature, there is quite a stark contrast in feature expression. That being said, this is just one of the many features working in tandem to differentiate chronic from acute antibody-mediated rejection, as well as other disease subtypes. And the takeaway message here is that analysis tools that incorporate spatial proximity and localization are capable of revealing novel forms of intra- and inter-microenvironmental heterogeneity. So in short, we have introduced an open-source pathologist-in-the-loop workflow for the quantitative assessment of microenvironmental heterogeneity using the spatial domain. We have demonstrated the applicability to a wide range of multiplex studies and support across multiple imaging modalities. And finally, we highlighted several publications from past customers ranging from cancer immunotherapy, preclinical drug evaluation, and multiomic characterization of tissue microenvironments. So on that note, I'd like to thank everyone shown here, but primarily the Fortis team, not only for coordinating this webinar, but for also generating some of the highest quality multiplex images I've ever had the privilege of analyzing. There's an old saying which we typically use when dealing with poor quality images with, let's say, overly ambitious analysis goals. Garbage in, garbage out. But working with Fortis, I have to say my experience is more like treasure in, treasure out. I am confident that the results that we have provided to our customers are truly reflective of the biological underpinnings of their dataset. And I hope that these results will further enhance their understanding of their patient's spatial biology and continue to be of further value to future customers, hopefully such as yourselves. And on that note, I'd be happy to take any questions, comments, and hopefully not concerns. Thank you.